Digestive Physiology, Lecture 7. We're now in the small intestine. We've done the esophagus and the mouth. We did the stomach. We did the accessory organs. Here we are now in the stomach. Good diagram right there showing mesentery, okay, and the different regions of the small intestine. Most chemical digestion and nutrient absorption occur in the small intestine. This is one of the things I keep emphasizing. Notice how things I emphasize a lot tend to show up in quizzes and exams. That's because I think they're the most important things. So remember, I did the extra credit on the last exam because 10 of the things I emphasized a lot, people missed a lot. So don't do that again, okay? Small intestine, okay, duodenum curves around the head of the pancreas, and that's because the pancreas is secreting things into it, only about 10 inches long. Retroperitoneal, along with the pancreas, makes sense. If the pancreas is retroperitoneal, the duodenum would be too, because they're connected to each other. Receives stomach contents, pancreatic juice, and bile. Neutralizes stomach acids with all the bicarb that gets secreted by the pancreas. Emulsifies fats. Pepsin is inactivated by the increase in pH. So pepsin is now not going to do any more at this point. There are going to be pancreatic enzymes doing the role of what pepsin did. The jejunum, remember the next 8 feet, kind of in your upper abdomen. Large, tall, circular folds, thick with muscle. This is where most digestion and nutrient absorption occur, right, in this middle part. Ileum, last 12 feet. This is kind of, the touch-up, do the finishing touches here. Has Peyer's patches, clusters of lymphatic follicles, in order to catch any bad guys that made it this far. Ends at the ileocecal junction with the large intestine. Look, ileocecal, what does that tell you? It's where the ileum joins to the cecum, which is the first part of the colon, or large intestine. So the surface area, these circular folds, the ridges that this diagram doesn't really show, there in the lab PowerPoint, there was a good one. Showed how the small intestine looks like it's got bunches of ridges, and covering those ridges are the villi, right? So the circular folds, the plicae circularis, are a centimeter tall. They have only mucosa and submucosa. Chyme flows through. Look at the diagram on the right. You can see how the chyme is going to have to swirl through that. And as it does, it slows down. And that gives it a chance to come into contact with it has to work its way through the villi, and then there are the microvilli on top. They're going to have enzymes on them. Villi, about 1 millimeter tall, have blood vessels and lacteals. Lacteal is where we absorb fats. Blood vessels where we absorb sugars and amino acids. Lipids absorbed by lacteals, sugars and amino acids by blood. Microvilli are only a micron tall. That's a millionth of a meter. Those are, you can't even see them with a regular light microscope. That takes an electron microscope to see. So the microvilli completely cover the villi. They're sometimes called the brush border because they're like the bristles on a brush, okay? They have enzymes that do the final stages of digestion. Now, these are not enzymes that are secreted and float around in fluid. These are in-place enzymes. They actually stay put. They're part of the microvilli themselves. So as the chyme swirls by, it comes into contact, and those intact, in-place enzymes on the microvilli do the final breakdown of the nutrients, okay? Damn, I talked over the transition again. Okay, intestinal crypts. So below the villi are these crypts, little openings that lead down into the glands, the intestinal glands. Absorptive cells, goblet cells at the base, rapidly dividing cells. Once again, that chyme is acidic. It's digesting your own intestine, so you have to keep recreating your own intestine. These cells last only 3 to 6 days, get sloughed off and digested, all right? And then you excrete them. Panath cells secrete antibacterial stuff. Okay, good. Duodenal, old-fashioned name, Brunner's glands secrete bicarb. Again, you've got to neutralize all that acidic chyme from the stomach. So you make tons of bicarb, and that's what the duodenal glands. Peyer's patches, lymphatic follicles that fight any bad guys that have made it this far. You secrete about 1 to 2 liters, 1 to 2 quarts of Gatorade, intestinal juice every day. Water and mucus, pH here is 7.4 to 7.8. So in the stomach, the pH was down around 3, highly acidic. Now that we make it into the intestine, all that bicarb is going to neutralize, and we're back to not only just normal pH, but we're actually a little bit alkaline now. Here you see close-ups of villi and the jejunum, and then here in the histology slide, you can see the duodenal glands down there. See them? Look closely. You can see those layers we've been talking about, the serosa, the muscularis externa. There are the duodenal glands. There are the intestinal crypts and the villi. Okay, that's the way they actually look. Which is not a zymogen. Trypsinogen, chymotrypsinogen, procarboxypeptidase, and enterokinase are all our zymogens. Pause the movie, figure out your answer, then move on when you're done. Enterokinase is what activated trypsinogen, turned it into trypsin, then trypsin turned the others into their active forms. So enterokinase is just an enzyme, not a zymogen. So moving things through, okay? Remember, there are 2 layers of muscle throughout the gastrointestinal tract, circular and longitudinal. So what's going to happen here is the muscles are going to contract. They have to mix the chyme with the intestinal juice and the bile and the pancreatic juice. It's like when you make something, you're making food, you're following a recipe. At some point, a lot of times you have to put stuff in a bowl and mix it all up. That's what's going on here. The churning actions of the muscles are actually mixing the stuff up, okay? Churns the chyme to increase contact with the mucosa for absorption and digestion. Moves residue towards the large intestine. We're going to be absorbing nutrients, but whatever we can absorb has to be eliminated from the body. Segmentation, this is where the circular muscles contract to squish the food, mix and churn the contents, 12 times per minute in the duodenum. Then the longitudinal muscles are for peristalsis. That is a linear movement. So it's going to squeeze the food along and move it down. So that diagram that's in the upper right, you can see on the right, so part B of that diagram is segmentation. Those are the circular muscles mashing up the food, squishing it up, mixing and churning it together. Then part A is peristalsis. That's where subsequent waves keep moving the stuff further and further along towards the exit. Push chyme along for about 2 hours. Suppressed by refilling of the stomach, all right? So once again, homeostatic mechanisms here, trying to keep everything well-timed. Food in the stomach causes the gastroileal reflex. Once again, just look at the name, gastroileal. Stomach talks to the ileum. So when food gets into the stomach, it sends a message to the ileum saying, hey, we got more stuff coming down the pipeline. There's more stuff coming down the assembly line. So it's time for you to start moving stuff towards the exit. So what happens, therefore, in the pyloric region, we will secrete contents into the cecum and start moving it towards the exit. Again, feedback throughout the digestive tract so that things move at the proper pace. The worst thing in your digestive tract is for stuff to get clogged up and stuck. So lots of hormones and signals, nervous reflexes that are keeping all this moving in a very efficient flow, all right? There you see segmentation, all right? The circular muscle, squishing it up. Purpose of segmentation is to mix and churn, not to move the material along. That's peristalsis. And here is peristalsis. So you can see now the diagram in the lower right. The contractions of the longitudinal muscles come in waves. So the first wave moves it a little bit along. Then the second wave starts where it is now and then moves it down further. Then the next wave starts there. So bit by bit, it keeps moving towards the exit. And notice there that girl wearing a shirt says, peristalsis, get shit done. One semester, students found one of those t-shirts and gave it to me. I would have worn it today if we were all together. Gradual movement of the contents towards the colon begins after absorption occurs, waves of contraction. Second wave begins distal toward the first, third wave distal to the second, and so on. Keep moving it down. And that's Torweep. That's where my soul lives. I've been going to Torweep since 1969, so that's over 50 years I've been going there. And every time I go there, I just do the same things. I just look at the rocks and the water and the clouds, and I just walk along the room and I just go, wow. Okay, I'll see you in Digestive Physiology PowerPoint number 8.